So good morning, everyone. Thanks again for joining, and I'm really excited to introduce our speaker today. But before we get started, just a few reminders. One, this is being recorded, so and we do post these talks on YouTube after the fact. So if in any event you don't want your face to be seen on YouTube, or you want me to say your name, that's fine. Just keep in mind that this is being recorded, and then it will be up on YouTube. So if you want to ask me a question anonymously, you can just send me the question directly, and I can ask it on your behalf. Ideally, what we've been doing is people put up their hand, or they chat me that they want to ask a question, and I'll try to find a spot so we can have a bit more of a free-flowing conversation during the talk. So with that said, I'd really like to thank our speaker, Aristeidis, for taking the time to give a talk here. I've been following his work specifically for a while. In our case, specifically the work on non-negative matrix factorization, but there's a whole host of other stuff that he does. He's an assistant professor at the Department of Radiology and the Institute of Informatics at Washington University School of Medicine in St. Louis. His research interests are in the field of medical image analysis, machine learning, and computational neuroscience. And he's very interested in developing novel computational methods to extract information from imaging data and to delineate patterns in large heterogeneous data sets towards improving patient-specific diagnosis and advancing our understanding of brain structure and function in health and disease. His most recent research work is based on developing machine learning techniques to address challenges in high-dimensional data, especially in neuroimaging and in disease heterogeneity. So in his talk today, he'll be talking about some of the most recently developed techniques and applications in large neuroimaging data sets. So once again, thank you very much, Aris, and looking forward to your talk. Take it away.
Thank you for inviting me to give this talk, and for the introduction. It's a pleasure to be at home and still be able to communicate and present some of our recent work. I would suggest that everybody mutes their mics, because I hear some return. Somebody hear myself on my headphones, so it's not the best thing. So the introduction was really good, but I always like to talk about myself. So as the name suggests, and I guess even the accent, I was born and raised in Greece, where I did my undergrad in electrical and computer engineering in the National Technical University of Athens. That's before moving to France and Paris, where I did my master and PhD in applied mathematics. And that took me then to UPenn, where I was postdoc first and then a research associate. So there is this west bounding trajectory, which took me a little bit more west and in the Midwest now, in St. Louis, where I'm currently assistant professor at the School of Medicine. As mentioned before, a lot of my work is to develop novel computational methods with the goal to extract information from imaging data and try to find patterns. And the ultimate goal is to improve patient-specific diagnosis and prognosis, and better understand brain structure and function in health and disease. So this kind of involves work in image processing, machine learning, and computational neuroscience. Briefly, I would say that I have worked a lot on image processing. That was kind of the earlier, my earlier PhD work. So I developed a lot of image registration and image segmentation techniques. This took me then to kind of, okay, we have extracted all this nice quantitative information from imaging data. We've processed these large data sets, how we can create new knowledge. So that required some machine learning development techniques. And there I focused on two challenges, the case of dimensionality and the heterogeneity. And my work has found application across different things, from brain development to aging and Alzheimer's disease, as well as brain tumors, and specifically brain tumor segmentation. Today, I will focus on the challenges of the dimensionality in the first part of my talk, and heterogeneity in the second part. And the applications that I will show you are on brain development. So there are two parts. If you want, you can jump in, I guess, a bit as we are changing from part one to part two regarding if you have any questions. I will be happy to reply as we go. So the first part of the work is how we can find, how we can reduce the dimensionality of our data in an interpretable way. And as I said before, this tackles kind of the challenge of the case of dimensionality. So what I was trying to illustrate here is that what happens as we are increasing the number of features for a fixed number of samples. So assuming here that we have 5 different features that we use to describe the brains, and we have 2 classes that we want to distinguish, the yellow and the red brains. So as we go from one-dimensional data to d-dimensional data, the density of the space really decreases. So we have a sparsely populated space. This gives us the opportunity to easily find ways and linear hyperplanes to distinguish between the different classes. But because of the fact that we have now such a sparse space, we can have we have millions of voxels and only a few, at best, thousands of samples. We can have overfitting, we can have spurious correlations, and we certainly have issues with multiple comparisons. So this requires techniques that will allow us to summarize in an optimal way the high-dimensional data. What we typically do, I guess, in most of the structural neuroimaging community, is to use a fixed atlas, or Kiliani or something else, to summarize the information from the thousands of voxels to a few hundreds of regions of interest. So that has the advantage that we have a high regional specificity. It is easy to interpret, but it's usually not adapted to the data. We have the same atlas that we apply across different studies, be it developmental cohorts or aging cohorts. And that might mean that this might not be the optimal representation of our data for this specific study. To find data-driven and study-specific representations, a lot of work has been focusing on multivariate pattern analysis techniques. And some of the more widely used are the principal component analysis and the independent component analysis. These techniques have the advantage that they are adapted to the data, so we get a data-specific representation, but they have some limitations that come from the fact that they model the data using both negative and positive loadings. So here on the right, I'm showing you 3 typical principal components that have been estimated from a study of aging, where we have given as input to the PCA gray matter tissue density maps. So you see that because now we are using both positive and negative values to represent our data, we have these complex mutual cancellations between regions of opposite sign. So you see, for example, the periventricular areas here are shown with positive values, as shown by the warmer colors. And in this principal component, they are having negative values, as denoted by the colder colors. So because of this mutual cancellation now, it becomes much more difficult to interpret the data. And because they are really greedy in their approach of representing the data, we often have problems of limited reproducibility. So our goal was to find ways of summarizing this data that will allow us to keep the regional specificity of the regions of interest approach, but they will still give us a data-driven study-specific representation. So we approached this problem in a few years back now, neuroimage paper, where we applied and explored non-negative matrix factorization for this problem. So the reason that we chose non-negative matrix factorization is because we were aiming to find representations of regions that are specific, the regional specific, easy to interpret, they are adapted to the data, and they are reproducible. And that our motivation was based on the fact that these non-negative constraints that we have in this matrix factorization lead to what is known as a part space representation. So I'm showing you here on the bottom an example where we have a cortical thickness map that is represented by in an additive way by a linear combination of different non-negative components. And now you see that these non-negative components, because of the constraints that we have applied, are really well localized, capturing different parts of the brain. So this is this is quite easy to interpret because we have these participant-specific coefficients, and we have parts-based components that are look alike a lot our standard anatomical parcellation. So it's a representation that has both good interpretability and allow us to do all the regression analysis that we are interested in. So how does non-negative matrix factorization work? So NMF, similar to PCA and ICA, are part of what is known as regularized matrix factorizations. So what we have here is typically data matrix X that has been constructed by putting side by side, column-wise, vectorized versions of our data. So here we have a cortical thickness map that we have taken, we have vectorized for subjects I, and we have put side by side all the data. So that gives us a data matrix X that has dimensions voxels times number of subjects. And what we ask the method to do is to give us now two matrices B and C, whose product will optimally reconstruct the data matrix X. So now the matrix B, which contains our bases and components in the columns, has dimensions voxels times k, where k is the user-specified variable that tells us how many non-negative components we are going to estimate. So in every column here, we have a non-negative component that groups elements in the brain that co-vary in a consistent way across the population. The matrix C now has dimensions k times n. So in every row of this matrix, we have subject-specific coefficients for the respective component. And we saw an example here on the bottom. So now B and C are constrained to be non-negative. So this is kind of the graphical summary of the method. This translates to an optimization problem. So what we try to do is to approximate the data matrix X based on the product of B and C under the Frobenius norm. We have the constraints that B and C need to be non-negative. Here we have a projective constraint on C and some orthogonality on our non-negative components. We can achieve this solution by using the multiplicative updates rules. This is a kind of a rescaled gradient descent that allows us to reduce the energy while respecting our constraints. And what is important is that we have used an initialization based on the non-negative double singular value decomposition. And this has the advantage that provides us with a better starting point. So we have a faster convergence, and more importantly, it's deterministic. So whenever we give the same data, we always get the same results. So in our initial experiments, we wanted to do a head-to-head comparison with PCA and ICA. To do that, we used data from the Baltimore longitudinal study of aging, where we have participants 50 to 85 years old. So here we use what are known as Raven's maps, which are gray matter tissue density maps. Essentially, we take the brain, we segment it into three classes, gray matter, white matter, and CSF. We deform it to a common atlas. And then by combining the information from the gray matter tissue class and the determinant of the Jacobian of the deformation field, we get a tissue density map that in every location of the template, it tells us how much we need to expand or to contract the gray matter volume of the participant to get to match it to the template. And then we did, using that as an input, now we vectorized these gray matter tissue density maps. We put them side by side, constructed our data matrix X, and we gave that as an input to PCA and ICA. And we did both qualitative and quantitative comparisons. First, let us see how the PCA and ICA components look for this data. So what I have here is two typical principal components on the top and two typical independent components on the bottom. So we have multiple axial slices, and what we saw is the components overlaid on top of our atlas, and we have warmer colors for positive loadings and colder colors for negative loadings. So what I would like you to notice is that the components that we are estimating are spatially dispersed. They overlap a lot, And we have these complex cancellations of regions of opposite sign. And that happens for both PCA and ICA. Now, in the same data set, if we see the non-negative components, we see that they're much more localized. They have great regional specificity, and they are much easier to interpret. Going now to some quantitative comparisons, we aim to quantify what is interpretability. And our approach for that was to use sparsity. So sparsity, higher sparsity means more elements of the component being equal to 0. Having to deal with less elements, usually it is easier. And what we did here is to evaluate sparsity by varying the number of components that we are estimating. So we go here from 2 to 100, and we evaluate representations for both, for all PCA in black, ICA in blue, and non-negative matrix factorization in red. So higher values means better. And what you see here is that ICA exhibits higher sparsity than PCA across all resolutions. What is interesting, though, is that non-negative matrix factorization is much sparser than both methods. And what is interesting to note is that as we are increasing the number of components, we are getting more and more well-localized solutions. So we're getting sparser solutions that capture better localized parts of the brain. Another aspect that we wanted to look into was the reproducibility of our representation. So what we did is we took our data, we split that in two halves. We evaluate, we estimate representations in the first half and in the second half. We match them, and we measure how much they overlap after establishing correspondence between the components. And we did that again by varying the number of components here from 2 to around 50. Again, results for PCA are shown in black, ICA in blue, and non-negative matrix factorization in red. So you see that all three methods are quite reproducible for a small number of components. But as we are increasing the number of components, the reproducibility of both PCA and ICA drops. And while the non-negative matrix factorization, after initial drop, it achieves a plateau and remains stable. This is because we believe that PCA and ICA are more greedy in capturing variations in the data. So at some point, they're starting modeling noise, and that limits their reproducibility. Whereas non-negative matrix factorization, because of the constraints that have been applied, it is a little bit more robust. On the downside, I have to say, is that the approximation error of the non-negative matrix factorization that I don't show here is much worse. It's much larger than the approximation error that you get from PCA and ICA. So I won't show you here any results, more details about the components we have estimated, because I will show you next a few slides from an application paper that we have done. What I would like to say is that if you have any question about the comparison, that might be a good time to ask now. Otherwise, I will move to the NMF application. I take that this is good. So that was our initial starting point. Since then, I think we have validated this tool a lot, and we have applied it to different settings, and it has led to a number of different papers. So I will just show you quickly that we have this is the paper that I'm going to talk next about our applications on the PNC and our approach of using NMF to map development there. But we have used it for pre-10 data and these exotic tensor derived measures. We have used it to in the PNC again to find associations between cortical networks and impulsive choice and gestational age. But and more recently, we also applied that on aging in our effort to kind of delineate the patterns of white matter hyperintensities. And we've also used that for age prediction. So what I would like though to focus on is our application to brain development, our first work on the PNC data. So here we use the non-negative matrix factorization to find patterns of coordinated cortical remodeling. And we checked links with function and evolutionary expansion. Our motivation behind this work was that while there is a lot of knowledge, a lot of studies that have been applied to development, a lot of them are kind of localization in nature. So we see here a well-known result on the left. So we see how the cortical thickness changes across age from 5 to 20, and we see that we have this spatially heterogeneous pattern. But a lot of this knowledge has been even on the vertex, on specific vertices, or on specific ROIs. So this doesn't allow modeling of this highly networked system that the brain is. So because they were using only this univariate type of approaches. So our effort, our goal was to use this multivariate pattern technique that will allow us to capture better covariations across the data. So in collaboration with the Brain Behavioral Laboratory and with Ted Satterthwaite, we used their data from the Philadelphia Neurodevelopmental Cohort that we had about 934 subjects that survived QC, and they were processed by FreeSurfer. So here we have cortical thickness maps. So yes, you know better than me that cortical thickness here is defined as the distance between the pi surface and the white matter surface. So now we have these maps for left and right hemispheres. We were using the FS average 5 template, so that makes it around 10K per hemisphere. We took the data for every subject. We vectorized left and right hemisphere. We concatenate them together and created the data matrix X that has dimensions around 20K times 934. And we gave that as an input to non-negative matrix factorization. So we did some analysis to find what is the optimal number of components. And the results that I'm going to show you now are focusing on what we found to be the optimal representation. So here I have kind of summarized all the components that we were able to find. So you see kind of the big brains below are a sort of a map. And we have placed the smaller components that show the patterns in locations that mostly correspond to the pattern that where the pattern has the higher loadings. So you have left and right lateral hemispheric surfaces, and you see most of the components that we estimated. Here we estimated, here is 18 solution. So I would like to go through some of the properties of the representation that we were able to find. First of all, we see that there is a lot of symmetry. So here you see in the occipital cortex, we have components that mirror each other. This is also the case for insula, frontal, and motor and sensory cortices. However, there are cases where we have asymmetry. And here is a good example where we have the Wernicke's and Broca's area. But what we liked and appreciated here is that the asymmetry happens in areas where we expect it to happen, where we have some functional lateralization. So this was something that boosted more our confidence about the biological plausibility of our representation. What I would also like to point out is that they are different from gyral-based atlases. And I like the example here that is shown that we have the temporal parcellation actually into anterior and posterior part. That's kind of distinct from what you will get from a gyral-based atlases, where they will split it into inferior, middle, and superior parts. And this is interesting because we see that in the trajectories, they do have different properties and different different age-related changes. So one is quadratic while the other one is linear, but also differences in the thickness. And these changes we would have lost if we would have averaged across the gyral-based boundaries. So what I would like also to point out that these components are spatially contiguous. And this contiguity is not driven by simply by spatial proximity, because we see that they are across hemispheres, and it's not driven simply by smoothness, because we have these elongated components in motor sensory and also in the cingulate that is not shown here. And the components that we see here, they speak for some functional importance of this representation. And this is something that was really of interest for us, and something that we further explored in our subsequent analysis. To do that, we used, to evaluate structure to function correspondence, we took the data from Yeo et al., the 7 functional networks, and what we did is to measure the overlap between our structural networks and the functional networks derived in another cohort. So we did some spin tests to find null distributions, and what I'm showing you here is the overlaps that are most significant. And I hope you can see kind of the pink boundary, the purple line here. This denotes the boundary of the functional network. And as a heat map, you can see our structural network. So you see for the visual cortex, we have almost one-to-one correspondence between structure and function. This was also the case when we looked at the limbic network. So you see kind of the purple line kind of enclosing the heat map that we were able to extract from this data. So interestingly enough is that in the somatomotor network was actually captured by 3 different structural networks here. So you see in purple the boundary, and you see how the 3 structural networks here almost are able to reconstruct nicely the purple boundary of the functional network. So as you see, as we are going from lower order to functional to higher order networks, we have the strength of this link is kind of diminished. So we have kind of a correspondence that is inversely related to functional complexity. So as we were moving to higher order networks, this was further diminished. So we were not able to find some strong correspondence with a default mode network, but we were able to find some structural networks having a good correspondence with parts of the dorsal and the ventral rotation network. So the next thing that we did with this data was kind of see how they change with age. And one important observation that we had is that a lot of these age-related changes seem to be driven or being explained by evolutionary expansion. And that had been discussed previously in the literature, but what was missing were kind of data there. So what we did is we took data from a paper published by Hill et al. In PNAS, where they had measured the evolutionary expansion by comparing a human brain to the macaque brain. And what we did is we looked inside the structural networks that we had estimated, averaged the evolutionary expansion, and we related that with the average thickness and the age-related change there. And what was interesting to see is that what we have here is that thicker cortex seems to be positively correlated with evolutionary novelty. What was more interesting for us was that the age-related change was negatively correlated, associated with evolutionary expansion. So we have this opposite direction here. This suggests that what is interesting is that what kind of makes us more human is actually this more dramatic change during this developmental period. With that, I will kind of close the NMF chapter, and I will tell you that I think hope I have convinced you that this is a tool that gives us nice interpretable representations. It is highly reproducible and versatile, so we have applied it to both neuroimaging data and also other non-imaging data. It does provide us with an optimal dimensionality reduction, but what I would like to say, it's not only this is not only a statistical contrast that allows us to boost our statistical power, but these networks seem to be biologically meaningful. And I will pause to see if there are any questions before I jump to the heterogeneity part.
I have a couple of questions.
Sure.
I mean, great stuff. It's super fascinating. I was wondering, first one, can you talk a little bit about how you choose the number of components? Because it's often a contentious issue when
yes.
Everyone has their own, like, you know, secret sauce for figuring out how many components to use. So I'd love to hear a little bit more about that.
Yeah, let me see. I'm going through my slide deck of slides to see if I have. Okay. So, and you can see, and this is the figure 1, I think, from this PNAS paper. So we typically, what we do is we look at the reproducibility. So a lot of our analysis and a lot of our intuition is that we want representations that are stable across splits, across validated folds. So we take, in one hand, we look at solutions that are stable, and we also take into account the reconstruction error. So we want a solution that is both stable but fits the data well. So here was the results from the PNAS. So our reproducibility analysis again was, we did a split half analysis and we evaluated solutions from 2 to 60. And you see that we have some clear peaks of reproducibility. And you also need to keep in mind that as we are increasing the number of components, the number of patterns that you are estimating, you fit the data better. So in our analysis, we discussed all solutions, all peaks here, the 2, 7, and 18, but we focused on the 18 component solution that is also fitting the data better. So it is a combination of how well I fit the data and how stable I am, either in a k-fold cross-validated sense or in a split half experiment setting.
That's great. I guess the other question I had is about the homology between, or the matchup between the structural results and the functional results.
Mm-hmm.
You know, I mean, I think that's really interesting, but but it's interesting you see this kind of nice match in certain types of regions versus, more kind of, yeah. So do you have a sense of if that's because of the type of features that you used as inputs, and if you had more complexity, that captured kind of various dimensions of the architecture, would that potentially do a better job and be lining up better with function? Have you examined that at all?
No, here, and I don't know, I always feel unconfident when I'm talking about neuroscience because my training is engineering and applied math PhD, so I do understand and but I'll try to tell you what this is my intuition, but the thing as I understand things is that these lower order networks are they have to develop earlier, so they have a more specific task, so they are kind of more specific. So it kind of makes sense that they correspond to structure well. By the default mode network that kind of does a little higher order task that needs to combine inputs from multiple areas, let's say from multiple types of information. I think it's kind of reasonable that it doesn't develop as one big chunk of structure, at least. I hope that I didn't covered you somehow, but
it was great. I'm going to have Lonnie first because she had her hand up first, and then we have a question from Casey.
Thanks. This is from the first part of the talk. I didn't ask it then, but I was just wondering if between the different methodologies you use, they make the same assumptions about like normality of the data and standardization. And I assume that you had like pre-processed them all the same way in that regard before running the PCA and the NMF.
Yes. Yeah, we did the same processing, the same assumptions, the same type of visualization, similar ways of coming up with thresholds to visualize the components. I thought that was kind of the most fair way of going, and that's what we have implemented.
And Casey's asking, Casey, you want to ask your own question, or you want me to do it?
Yeah, I can ask. Thanks, Aris. I was wondering, sorry if I missed this, but is there a way to put multiple features into the non-negative matrix factorization so it can act as some type of fusion?
Yes, so we are working on that now, so yeah, it is feasible. I would say that. I think we haven't published anything yet, but it's in the submission of my R1, so I'm working on that right now. But I can tell you that another interesting usage of the NMF that we have done is that we have defined, for example, patterns based on one population, and it is possible to apply them to another population. So if you want to see how these structural networks how well they fare, I don't know, in area or gyrification, it is easy to take them and take the new data set as well as they are spatially aligned as soon as well, provided that they are in the same space. You can project them to the representation that you have estimated and calculate subject-specific coefficients and do all your analysis. So,
okay, cool.
If I'm going to be
and
totally,
thanks for that. Thanks for the link as well.
We've done that work. Rayhan's that work in his PhD.
Yeah, I thought it sounded familiar as soon as I asked the question.
Okay, I'm not trying to
plug.
It's not the place. We have one more question from Torgir. Do you want to ask it? Do you want me to ask it? I'm happy to do it. I will ask it. How dependent are? Oh no, he's unmuted himself.
I'm on. So yeah, in my own data, I've seen something a little puzzling. So I've had a very large data set and then just split half. And for lower model orders, I get very similar solutions across split halves. But then when I run it on the whole sample, I get a solution that's slightly different again. So I'm just wondering how dependent the solution is on the number of data points.
So, and I guess you can see it here. Let me pull that again. Certainly, as you are increasing the number of components, I think you should expect that the reproducibility will be decreasing. Yeah, and we saw it here. I think that's, so you see, when you have two components, they are almost the same. So they are mirroring each other. So they are almost identical. As you are going, as you are increasing the number of components that you're estimating, it does drop. But I think where it drops, it remains quite high. I think that's a common property that you see it everywhere. I think with every clustering, let's say, technique. I think that something similar, for example, it was also reported by Yale in their functional organization paper, where they were using a clustering technique to find the 17 functional networks. So this is something to be expected because the complexity is increasing. Have more degrees of freedom. There will be standardly more variability. So here I was using the mean inner product or the median to evaluate reproducibility. So there is certainly a bias towards a lower number of components. Since then, I have been also experimenting with the adjusted rand index that takes into account takes into account this challenge. And then you will see that actually it gives you a little bit the opposite view. So if you account for the chance of grouping two things together, which is higher for smaller components, for a smaller number of components, then it gives you kind of an opposite profile. It gives you a highly stable adjusted rand index for a larger number of components. So I think taking both the inner product and the adjusted rand index into account, I think it points that the representations overall are really stable.
Yeah, just to follow up a little bit, because what was puzzling for me was I got for two splits for 2 components, 3 components, 4 components, they were almost identical. So
yes,
but then I asked for 2 components, 3 components, 4 components in the full sample, and I got something different. It's not totally different, but different. So I would expect that. So that's why I'm wondering sort of how sensitive are they to the initial number of data points that you give out.
I see that sounds okay. So I mean, it is a data-driven representation. So it's trying to fit your data as best as possible. So so I would expect some variation, but if you have a large sample, I would expect this variation to be smaller than not so significant, and I guess this variation will be becoming more prominent as you are as you're going to higher resolution. So for the solution of 2 or 5, 2 to 4 components, I think it should be really close to each other. But as you're going to, I don't know, 18 component solutions, you should expect some drop. I think that's kind of the power of these methods, that they are data driven and study specific. So they do, they are influenced by the data that go inside. Yeah, I guess that's also some sort of weakness if you won't get the same thing all the time.
Thanks.
Okay, good. We guess we can move on.
Okay, I'll jump and I'll try and see to keep some things to maybe move faster. So the second thing that I've been working on quite a bit lately is the heterogeneity. So, and specifically in brain diseases. So we know that a lot of these disorders are heterogeneous. So we know that Alzheimer's or schizophrenia, while they have the same clinical variable, they are categorical variable, they hide a lot of variability within them. But despite this, the fact that we know that they are heterogeneous, a lot of the tools that we are using are trying to find a single imaging pattern that will distinguish between the healthy controls and the disease population. So that is true for the standard voxel-based analysis that we're using, or the standard support vector machines classifications that they're trying to find a pattern that distinguishes between healthy and schizophrenia population. So while we have recognized that there is this heterogeneity, our analytical tools may not be get well adapted to tackle this problem, and they tend to give us an incomplete representation of the differences between healthy and disease populations. So I'll try to illustrate that here, where we have this control population showing by these dark circles, and we have this heterogeneous patient population that is denoted by the different shapes. So now if you would do your support vector machine and you will try to find a linear discriminant that will distinguish between these two populations, you would find maybe a line like that will capture kind of the common, the central tendency of these differences between healthy and disease populations. So that will give you kind of the common denominator of the differences that exist. So this is only but this is only capturing part of the of what is happening here. Ideally, we would like to be able to find that there are different subgroups and to estimate the different different patterns and different that distinguish the healthy controls from the different disease subgroups. So this is I guess it's becoming more and more important, and people are paying more and more attention to that. And here I'm using a figure from Incel Science paper, if I'm not mistaken, and where they argue that we want to go away from the symptom-based categories, and we want to deconstruct them, put all the data together, use all our available information now to reconstruct data-driven categories that are more homogeneous. And the ultimate goal of that is this will lead to better understanding of the brain mechanisms. It will allow us to do better diagnosis and prognosis, and might have implications for therapeutic innovation, because it will allow us to do targeted recruitment for clinical trials. So in this context, there are some, I guess, at the point when we did, it's kind of expanding literature, and more people are paying attention to that. But initially, I guess the first approaches were trying to find subgroups by using pathology or other clinical evaluations to define these disease subgroups. And this has the limitation that some of this information might be hard to acquire, and it is based on it is noisy, it might be noisy, and it is biased to our understanding of what is pathology and what is not. And there are data-driven clustering approaches, and I think this is mostly the literature that I'm citing here is mostly pertinent to Alzheimer's disease, where they have been using mostly hierarchical clustering or K-means. And these naive, unsupervised clusterings have the limitations that are affected by confounding variations. So they tend to be influenced by sex or age similarities. So they tend to subgroup the population based on older and younger people, for example, which is not what we would like, as it's not aligning well with underlying subtypes of pathology. So aiming to tackle that, we came up with the PhD student that I was working at that time, Erdem Varol, and with Christos Davadzikos, we proposed HYDRA. We really like the name and reference to the Greek mythology, which stands for Heterogeneity through Discriminative Analysis. So what we try to do here is to do an extension of the support vector machines and try to do both clustering and classification at the same time. So our idea was to extend support vector machines from the linear case to the nonlinear case. And instead of using kernels to create this nonlinear boundary, what we did is we created this nonlinear boundary that you see here in green by piecing together multiple linear segments. That allowed us to construct a convex polytope that puts in one side of this half space the healthy control population and in the other side the disease population. But it allows it allows us to do classification. But what is interesting, it's also allowing us to do subtyping. So it allows us to do clustering by assigning parts of the disease population to the faces of the polytope that best distinguishes them from the healthy controls. So in that sense, now clustering is not driven by how similar these subjects are with each other, but instead is driven by how similarly different, if you want, they are from the healthy controls. So I guess, I don't know, seeing the time, I think I will skip the math and I will tell you that we have been using this. So we validated this method initially on Alzheimer's disease population, but we have been using it lately to parse the heterogeneity in schizophrenia and in use with internalizing symptoms. So I will focus on the latter paper. So we'll see again an application to brain development. The work was led by Tony, who was at the time postdoc with Ted Satterthwaite. And here again, we looked at the Philadelphia neurodevelopmental cohort, and we tried to see what is the heterogeneity in use with internalizing symptoms. So our kind of the motivation was that there is high comorbidity among disorders. They saw similarities, but they have heterogeneous clinical presentations. So, and the DSM classification, as I guess more and more people are talking about, might not be the best tool to capture all these things. So the purpose of our analysis was to try to use HYDRA to parse the heterogeneity, as this is associated with youth that has internalizing symptoms. So we started with the PNC data. So we had about 1,600 people that had undergone structural imaging. We did standard exclusions for quality assurance or excluding people that have medical disorders, some missing data. And that led us to have a final sample of 1,400. So a requirement for HYDRA to work is that you always need to have a healthy control. You always need to have a reference population, and then you try to find the heterogeneity on what you consider as disease. So here we create the disease population, our internalizing symptoms, by grouping everyone that had any sort of PTSD, panic, phobias, or different types of anxiety. And then we had, which was around 700 participants, and we had this typically developing youth of 400 participants. What we did is, as we said before, we focus on structure. So we used multi-atlas segmentations to estimate volumes around in 112 regions in the brain. And we also used cortical thickness, where we had 98 cortical thickness regions. These were processed by with the ANS pipeline. We corrected for age and sex by including the MES covariates in HYDRA. And then we asked the method to tell us how many homogeneous subgroups it can find. So that is a strategy that involves analysis of running the methods by asking multiple questions. So asking, can you find 2 subtypes, 3, etc. So we evaluated from 2 to 10. We have embedded in our analysis a cross-validation setting that happens behind the scenes. And by estimating now these multiple solutions, we evaluated their reproducibility in this cross-validated setting. We used the adjusted rand index. And as in the NMF case, we used the most stable solution that we analyzed further. So here we had a clear pick for 2 subtypes, which was, we did a null permutation to find some associated with p-values. It was statistically significant, and that's what we used for the rest of our analysis. So we have subtypes that were relatively balanced in size. So we have 403 participants belonging to subtype 1, while about 300 belonging to subtype 2. We had I think a greater percentage of females in both subtypes, corresponding to the typical developing youth that were balanced on sex. We didn't have age effects. I think maybe the subtype 1 was a little bit older than typically developing youth, but no other effects were significant. And then, having established these subtypes, we try to make sense of them. So to do that, we used in oral statistical analysis, you use the model that you see here. So we have a brain measure. We use the generalized additive model. So we have some nonlinear effects of age. We use the age and sex, as well as measures of data quality. And we have the groups as were provided by HYDRA, and we're asking questions how different they are, the groups between each other, and how different they are from the typically developing group youth. So our first conclusion, and I would caution you here that you shouldn't really see p-values and pay too much attention, because to things like p-values when you're using the same input features as the ones that you use for HYDRA. So we saw that the subtype 1 has smaller volumes, and you see here on the left slices, axial and coronal slices, so color-coded by the T statistic. And you see here the total gray matter volume for typically developing youth, subtype 1 and subtype 2. This is the mean, and this is the mean with 95% confidence intervals. So you see subtype 1 shows us that smaller volumes. If we go to the cortical thickness now, we see again the similar idea, with the exception of some regions, parahippocampal regions. So we have again the subtype 1 showing us reduced cortical thickness compared to both typical developing youth and subtype 2. So I caution you that you should always look at the input features that you give to the machine learning thing to to understand what your method did, but is to be we will have here. So to better validate and understand what was happening, we used data that were unseen, that were not used during the clustering approach. And we used both diffusion data, DTI data, and functional data. So first, we evaluated the differences by using low frequency fluctuations. And again, the figures are all the same setting. You have some visualization on the left, and you have some summary statistic on the right, and showing values for typical developing youth, subtype 1 and subtype 2. So we've seen that clusters that were defined based on neuroanatomical differences, based on volume and thickness, they show also differences in function. So we have reduced amplitudes of low frequency fluctuations in frontal regions and in the amygdala. If we go also to the TTI now, we see that again, the subtype 1 has a reduced FA in a number of tracts. So, and it is typically the same kind of pattern everywhere. We see that the subtype 1 is doing a little bit worse, while subtype 2 is doing a little bit better, or similarly to the typically developing youth. To further analyze the data, we took into account the cognition and psychopathology. And now again, you have typically developing youth, subtype 1, subtype 2, confidence intervals, only the mean for the typically developing youth. And we have here executive function, social cognition, and executive memory. So, and the same pattern, subtype 1 shows poorer cognitive performance. So we see that there's this pattern, it's always the same. The green one is doing worse, the typically developing, and subtype 2. What is interesting, for some of these tests, subtype 2 is not doing statistically significant different from the typically developing youth. So beyond the cognition, then what we checked was the psychopathology. For that, we use the Bifactor model that is the favorite in PNC. So we took all the items from the Go Assess interview. We did the Bifactor. We have, and we've got factors for anxious misery, psychosis, behavioral, and fear, and an overall psychopathology factor. And again, now we use this information to see how different are the typical developing youth from the different subtypes. Again, similar to what we saw in cognition, we have subtype 1 showing more psychopathology, overall psychopathology, as you see here. And we still have differences in behavior and fear estimate. So across the board, subtype 1 is doing worse than subtype 2 and typically developing youth. And I have to note that there was no significant differences between subtype 1 and subtype 2 for the anxious misery or psychosis factors. So I went a little bit fast here for due to time constraints, but our summary here is that HYDRA was able to find two subtypes of internalizing youth, one that has kind of smaller and thinner brains, while the other one has larger and thicker brains. Subtype 1 was more vulnerable. It had lower socioeconomic status, more psychopathology, reduced amplitudes of low frequency and FA, while subtype 2 was more resilient. I guess what will be the key message is that this categorical standard analysis that we are using might not be the best way of analyzing such type of data. And machine learning tools can delineate homogeneous subtypes. And that's what we were able to do here based on the brain structure. With that, I would like to acknowledge contributions from a lot of people, but here the material was specifically with Tony and Ted and with Christians and Erden. And I would like to thank you for your attention, and I would be happy to answer any questions that you have to this second part that went faster and more packed, I guess.
Great talk. Thanks so much. Any other questions from? We have time for, well, we don't have time for 5 more, but we're going to make time for 5 more minutes of questions. If anyone's got anything. So I have a question about the type of NMF, the variant of NMF that you're using all the way through. It looks like I didn't explicitly stated it in the talk, but you're using the orthonormal variant.
Yes, so I mean, orthonormality, there is orthonormality. I think the most interesting is the projectivity constraint. The orthonormality, I used it because it gives you an easier update rule. So it lessens kind of the computational burden. It's easier and it's a little faster. But in my experience, and I experimented a lot and I tried different things, and before converging to that, is this projectivity constraint that it makes a big difference. So the thing is now, because you have this projectivity constraint, you have let me think what's the term. You have a distributed representation. So all the factors are used to reconstruct every participant, and that has the results in not having factors that replicate. So with other variants where C is a free and estimated independently, you have factors that replicate, so you are more prone to overfitting, and you lose a lot of the interpretability that you would hope. So that makes also more challenging the task of finding the optimal number of components.
Anyone else have any questions? All right. Well, if you'll unmute yourself and thank me, join me in thanking Aris for such a great talk and really taking the time to come do this. I really appreciate it. So thank you very much. Really appreciate this, and hopefully see some of you guys back here for next week. All right, take care. Enjoy the sunshine.